These specimens, these are all from market weight pigs that were processed in the meats lab in the butcher block. And we just got these as some tissues that we can use for dissections. I thawed them last night. So this is a set of lungs and a heart from a pig. This is the trachea up here, got the esophagus, and here are our lungs. And this will set in the thoracic cavity like this, okay, with the heart kind of facing forward, sternum, rib cage would be up here, okay. Not a lot of space for the heart, right? It's all kind of compressed around everything there. So here's our heart, kind of open the lungs up a little bit, and the heart sits inside this pericardial sac, which is composed of the parietal layer of the pericardium. Visceral layer of the pericardium is right on the heart surface. It's nothing you can dissect away. Okay? So this makes a bag or a sac that the heart sits in. And there's always some fluid in there, 10 to 50 milliliters of fluid depending on the animal size and species. But the role of that fluid is to help lubricate the heart and allow it to move really freely without abrading against any of those surrounding tissues as it beats inside the thoracic cavity. It's all going to be kind of pressed together like that. Okay? And that fluid helps just lubricate and allow it to move really freely in that limited amount of space. If I take it out of the sac, okay, here we can see the auricles. Okay, so these are extensions of the atria, the upper chambers of the heart. Also, our major blood vessels are all up here. The aorta, pulmonary artery, pulmonary vein, vena cava are all located up there. This part is called the base of the heart. See ya. And this is the apex down here. So I think base and apex, a lot of people will turn that around on an exam. So apex has the point. I think the apex of a pyramid. Big blood vessels base is the other way to think about that. All those B's kind of fit up there, okay, on that side. Just help orient yourself. Okay. Then I've taken these hearts and I've cut them in different ways so we can look at the internal features. So here, I've just cut into the ventricle just to show, look at all that thick muscle. I mean, that's almost all muscle that makes up the ventricle. Okay, here's the internal surface of the ventricle. It's shiny, that's where the endocardium is located. This white fiber, okay, this is one of the chordae tendineae, so that attaches to the flaps of the atrioventricular valves and prevents those valve flaps from opening back up into the atria when the ventricles contract. You can see them better over here in these sections. I'll get to those in just a sec. Okay. The other thing is, and I'll come back to it when we talk about the electrical conductivity, okay, these are the upper chambers. Okay, this is cardiac muscle. These are just blood vessels. That's why I'm pushing them out of the way. So that's cardiac muscle. Okay. This is the cardiac muscle that makes up the ventricles. A lot more muscle. Okay, a lot more mass there to deal with. Okay, and that'll become important when we think about how did the impulse to undergo contraction, how is that trafficked, and when do the atria and ventricles contract, and why is that? So that we can use the heart as a pump to move blood out through the body. Okay, so I'll come back to the muscle in a little bit. On this heart, okay, I've cut it open into the left to show the left ventricle on this side, and I've cut it open on this side to show the right ventricle. Okay, and I know that the landmarks that I use to know that this would be the left ventricle, the opening of the ventricle goes all the way down to the apex of the heart. Okay. Real thick wall of muscle here on the external surface. Compare that to the right side, where the chamber only goes about 2/3 of the way down towards the apex. It doesn't go all the way down, and there's a lot less muscle in the wall. Okay. So thinking about the flow of blood, if I take my wand, I'm just going to go up into through the AV valve. So right now, my Explorer is pointing into, or is inside of, the right atrium or the right auricle. And you can use the terms auricle and atrium interchangeably as far as the lab goes. The auricle is just part of the atrium, but if you say either one, that's fine. I'm not going to worry about that. There's a major blood vessel that attaches here to the auricle or to the atrium, and this is on the right side. Okay, which blood vessel do you think that is that attaches to the right atrium to bring blood to the right side of the heart? What would that be?
Vena cava.
Yes, the vena cava, very good. So the vena cava is the major vein that brings all the deoxygenated blood back to the right side of the heart. And then from there, that blood will move from the right atrium into the right ventricle, okay, going through the right AV valve. Okay, these are the chordae tendineae and the flaps for that right AV valve right here. Okay. So as the right ventricle then contracts, that's going to force that AV valve to close. Pressure is going to keep building up in that right ventricle. And we're going to then open up the semilunar valve, okay, to force blood out of this vessel, to take blood to the lungs. So what blood vessel would this be? Pulmonary artery, that's right. So that's going to bring our deoxygenated blood over here to the lungs. It'll get oxygenated. It's going to come back to the heart. It's going to come back on the left side and enter into the left atrium, or left auricle here. What blood vessel is bringing it over to the left side of the heart?
Pulmonary vein.
Yeah, pulmonary vein. Okay, so veins always bring blood toward the heart. Arteries will take it away from the heart. Okay, so our blood enters then the left atrium. It's going to pass through the left AV valve where my finger is. Okay, and then fill up the left ventricle. When the left ventricle contracts, that's going to force the closure of the AV valve. Okay, and then the blood's going to be ejected through the aorta, which is right here where my Explorer came out. Okay, so that's how that blood will move. On this heart, you know, I said right ventricle cut through the septum, left ventricle. So this is our septum. That's just what divides the left and the right sides. We want to think about this area. This is where the atrioventricular node is going to be located, or the AV node, and I'll come back to that. And on this heart, when we're on the right side, okay, where the vena cava is located, this is where a cluster of spontaneously depolarizing cells are going to be located that set the pace of the heart. It's the pacemaker of the heart, called the SA node, sinoatrial node. It's not a structure we can morphologically identify, and we don't even have the part of the vena cava here, so you can't see it. It's not present, but it's not something I would ask you to identify as a structure. But that's going to bring they're going to spontaneously depolarize in that they don't have the ability to maintain a stable resting membrane potential. So they're going to depolarize. And then norepinephrine and acetylcholine are neurotransmitters that can influence the rate at which the SA node depolarizes. But SA node by itself just has this ability to keep depolarizing. As it depolarizes, it's going to send that impulse to all these cells that make up the upper chambers of the heart, because all those cardiac muscle cells are joined together by the intercalated discs, where we have gap junctions that allow the cytoplasm to all to be in contact, so that impulse will spread across the atria uniformly, causing all those cells to depolarize together, and then all those cells will contract, so the atria will contract together. Okay. The way we have a connection to the ventricles is by something called conducting fibers. So running from the SA node, embedded in the wall of the atrium, are these fibers that are conduits or pathways that route that impulse here to the AV node, that point in the septum of the heart. Okay, and so when those conducting fibers stimulate the AV node, that causes them to depolarize. Okay, so that impulse then is sent down the septum through something called the bundle of His, or the AV bundle, which then branches off into what are called Purkinje fibers that are going to be embedded in all this thick muscle that makes up the ventricles. Okay. There are intercalated discs that connect all these cardiac muscle cells in the ventricles together, but the intercalated discs don't connect the cells of the atria with the cells of the ventricle. So it's like we have upper chambers that are connected, and we have lower chambers that are connected. The only way the upper and lower are connected to one another electrically is by the structure called the conducting fibers. Okay. Now, the conducting fibers are important not only because they're the source of connection to the bottom chambers, but they also slow down the impulse that was generated by the SA node by about 100 microseconds. Okay, so that means the atria are going to depolarize a little bit before the ventricles depolarize. So the atria are going to contract a little bit before the the ventricles do. And that's important when you think through those pressure differences that I talked about in lecture and in PowerPoint about why do the AV valves open, why do they close, why do the semilunar valves open, why do they close. It's all about the pressure differences between the chambers, or between the two spaces on either side of each of the respective valves. So if we offset when the atria and the ventricles contract, okay, that allows us to have those pressure differences so that blood only flows in one direction. Okay. If the SA node was damaged and not able to generate action potentials anymore, the heart can still contract. It's because the AV node has the ability to spontaneously depolarize, so it can become the pacemaker of the heart. And when it does so, it'll depolarize, it sends its signal down the AV bundle, Purkinje fibers, just as if it would have if it originated in the SA node. The difference is we're only going to be allowing then the muscle cells that make up the ventricles to depolarize and contract. The atria won't be able to contract. The impulse is not going to run backwards, it only runs forwards. So in that case, you might think, well, how do we get blood from the atria down to the ventricles anyway? In reality, these chambers are really small, the atria. And so as long as the AV valves are open, blood is going to be returning, okay, via the vena cava or pulmonary vein and flowing into the atria. It's just going to flow right into the ventricles. And when the ventricles have a signal to contract, okay, they're going to start to contract. That's going to reduce the space, increase the pressure inside the ventricle. That'll force closure of those AV valves. Okay, and then whatever blood is trapped in the ventricles will be forced out through the aorta and pulmonary artery. So we can still get blood moving to the lungs, still get it moving to the body, but it just wouldn't be as effective because we don't have the atria really participating in the movement of that blood. Okay, so there would be symptoms, it would be known, but that doesn't mean the heart would completely stop if the SA node was damaged, because that AV node has the ability to still contract. If, though, on the other hand, I had an experimental animal and I used like a laser or something and I went into the AV node and I ablated it, destroyed it, or I destroyed the AV bundle in some way, well then even if the SA node is still functioning, there's no way to send that signal to depolarize to all the cells of the ventricle. Okay, so it would be unable to act as a pump. So the animal would die at that point if there was no ability to move electrical current through those cells of the ventricle. Questions?
What are the functions of you didn't talk about them in the PowerPoints, but they were like labeled like the little holes, like trabeculae. I don't know what they were called exactly, but like these things, like the divots, the circles.
I don't know. The papillary muscle would be in this region. I don't recall. I'm not sure.